So welcome to the University of Michigan Biological Station Seminar Series. For those of you that have not met me yet, I'm Amy Klassen and I'm the director of UMBS. We're thrilled you have all joined us tonight, both virtually and for those of you in watch parties lucky enough to be on the shores of Douglas Lake. The University of Michigan Biological Field Station, which exists on lands once occupied by Burt Lake Band of the Ottawa and Chippewa people. In coming together this evening for this seminar, we respectfully acknowledge the original inhabitants and the descendants of the land we now manages for the purposes of preservation, research, and education. We ask our community and the visitors here to respect the integrity of the land that was home to Indigenous people before our state and university were established by acknowledging their gift to us. This requires that our programs of research, education, and outreach all focus on the connections of all people to other living things and to our land, water, and air. So tonight I'm excited to welcome you to the Bennett Lecture in Plant Biology and Mycology. This lecture is in honor of Ralph E. Bennett, who was a student and a botany instructor at the University of Michigan Biological Lab. Dr. Bennett worked on a diversity of projects, including the early development of penicillin and other antibiotics. So he clearly worked in pioneering areas important for humanitarian purposes. He worked at a variety of universities, commercial institutions, and research institutions, and ended his career with a large number of accolades for his body of research, including becoming a fellow of the AAAS, a fellow of the American Academy of Microbiology, and a fellow of Sigma Xi, just to name a few. In honor of his distinguished service and research career, we now have the Bennett Lectures at the University of Michigan Biological Station. Like Dr. Bennett, Dr. Luke Nave is a researcher at UMBS who works at the interface of botany and mycology. He's an associate research professor at the University of Michigan, and lucky for us, has worked at the University of Michigan Biological Station for around, I think, 14 years. He received his PhD from the Ohio State University in 2007, and his bachelor's before that from Wittenberg University. He conducted his PhD work and his postdoctoral work at UMBS and continues to conduct field-defining work on forest ecology and soil organic matter dynamics at the station. Like Dr. Bennett, Dr. Nave's work is important to society. His work informs managers and policymakers on how best to manage our forest resources and think about carbon sequestration over time. Dr. Nave has collaborated with tonight's speaker on forest ecology and soil carbon dynamics. Given Dr. Nave's research prominence in the area, his working relationship with tonight's speaker, and his ability to dig soil cores to greater than 60 centimeters, I hear, which is pretty darn impressive, there's no better person to introduce tonight's speaker than Dr. Nave. So thanks, Luke, for giving our introduction.
You are too kind, Amy. All right, sharing my screen here. All right, do you see a slide? Okay. Amy, thank you for illuminating us all with that history of Dr. Ralph Bennett and the lecture endowed in his honor. It's my honor to introduce this year's speaker, Dr. Knute Nadelhoffer, Emeritus Professor of Ecology and Evolutionary Biology at the University of Michigan and Director of our beloved biostation from 2003 to 2020. I feel some sadness over doing this through a webcam when so much of the richness of Knute's character is tangible in person. But these are the times we're living in, at least for now. Knute has been many things to me. In no particular order, he's been my science idol, my mentor, my advocate, my close collaborator, and my friend. I've been fortunate to know him for well over 15 years, and knowing him through these years leads me to the same words about him that many others have spoken. Knute is kind, enthusiastic, honest, generous, Midwestern, unbelievably creative, extremely intelligent, and larger than life. That last one is so true, and it's the basis for my title slide here. There's enough to Knute in body and being that he sums up to at least 1.5, maybe a full 2 on some counts. Knute is a man who has never been mistaken for a polar bear. But he did once have a polar bear mistaken for him. Ask for the story sometime, preferably in person. Knute is a man who has traveled by Yak, Soviet military Jeep, and Boeing 747, all in the same day. Ask for the story on that one too, but don't be surprised if it runs on into 2 or 3 more. In life, we sometimes meet those rare people who seem to be sought out by good stories and unusual experiences. Knute is a man who had a bright career as a college defensive end before he decided to pursue science. And he once summed up his time in football in these words: "I didn't get head injuries, I gave head injuries." And I hope that his wife Barb is rolling her eyes just the same way right now as she did the time that I heard him say that, because it'll prove some things never change. But you know what? The proof that he's right is his science. If all those head injuries that he gave did anything at all, they only pushed Knute out of the box and into a realm of creative thinking that most of us can only admire. If you don't have your nose in topics like fine roots, below ground carbon allocation, and forest nitrogen cycling, you may not realize that in his 8 lead authored papers on those topics alone, Knute has been cited 2,000 times. Never mind that he's worked on many other topics across several major biomes on an uncounted number of projects. And if you're not impressed with those 2,000 likes by the scientific community, know this: as a lead author or co-author of over 160 journal articles, reports, book chapters, and data sets, Knute's thinking has been cited a whopping 34,000 times by scientists. No wonder over the course of his career, he's been selected for a Fulbright, elected as a fellow of the AAAS and ESA, and served in advisory and executive capacities for a variety of science and advocacy organizations. Nearest and dearest to my heart, or at least my professional interests, Knute built the foundation over 30 years ago of what we still think we know about the invisible yet massive role that fine roots play in forest carbon and nitrogen cycling. He didn't stop there though. This evening, I hope your ears are open and your attention is undivided, because Knute is going to take us on the wild ride of what I think may be his dearest professional obsession: spinning tales of soils, detritus inputs, organic matter turnover, and if I know anything about him, probably some funny experiences too. This guy, who we know and love each in our own ways, has a brain, a big brain, but for all it contains, it's not as big as his heart. Now, as the bumper sticker says, folks, it's time to get in, sit down, buckle up, and ride. All yours, Knute. Thank you.
Well, thank you. Thank you, Luke. I'm going to share my screen here. Am I muted?
Nope, you're good.
Okay. And my screen looks okay?
It looks great. Thank you.
All right. Thank you. So, Luke, thank you for the kind introduction, and Amy as well. I really made that football comment in jest. I hope I never gave any head injuries. I probably suffered some, and if I have, maybe that has, moved my brain around to develop some of the creative characteristics that Luke cited. I'm flattered and I am embarrassed at the generous introduction. Truly are a valued colleague, Luke, and Amy, I'm very pleased that you're the new director of the biological station, so thank you. So every good talk has to start with a New Yorker cartoon, and this is my favorite New Yorker cartoon ever. When dirt ruled the earth. Uh-oh. Okay. It still does, at least in part. But before I talk about dirt, I want to talk about my amazing collaborators. We began the dirt experiment, which I'll explain in a few minutes, in 2004. And in 2019, these were the active collaborators who are sharing their ideas, their expertise, their labs, and their hard work and creativity. The kinds of science we do in ecosystem ecology and biogeochemistry requires teams of people because there are just too many aspects to understand by oneself. I'll start in the upper left. Jim Lemoyne joined my lab in 2004, and the first thing he did was set up this dirt experiment with me, which I'll explain. He left for, he now is at Northern Arizona University, but he came back in 2019 on his own time to help us sample. So every time this, you see a data slide, you can rest assured that the sampling was done in exactly the same way because Jim Lemoyne never forgets anything. John Denial succeeded Jim as my lab manager when Jim left for another job. And John has carried on and done the work on the DIRT project and has provided many of the graphs that you'll see today. Brooke Propson was a field assistant with us. She is now starting a PhD program at the University of Wisconsin-Madison, where I earned my PhD. So I've given her lots of good hints about how to survive in Madison. Isla Wrightson and her PhD student, oops, these are reversed. This is Professor Myrna Simpson from the University of Toronto. Her student, Isla Wrightson, has produced some data which I have in my pocket, but I probably won't have time to show today. Professor Kate Loida, a long-term friend at Oregon State University, has played a key role in coordinating the DIRT network across the Northern Hemisphere. And she's been a great collaborator. I'll explain more about how that works. Professor Sarita Fry at the University of New Hampshire. She is an expert in microbial ecology, and we rely on her to analyze our soil biological, mycorrhizal, fungal, and bacterial communities. Professor Rich Bowden was a postdoc at Woods Hole while I was there, and he got the idea when he became a professor at Allegheny College to start a DIRT experiment there. Rihanna Lucas, Olivia Tu, and Julian Mallerby joined us on our 2019 sampling. You see their picture there. And I want to thank everyone at UMBS. We've had scores of students working in the field, several REU projects, research experience for undergrads, several class projects, people collecting data. Some of our graduate students have studied processes at the DIRT sites at the University of Michigan Biological Station. I want to give a particular shout out to Helen Habicht, the new chemist at the Biological Station, who basically started her job in June in the midst of the pandemic, had to pick up a lab, repair equipment, get equipment serviced when the supply chains weren't really operating well. Many of the data I show today just came into my mailbox about a week ago. And I've been working almost around the clock to interpret these data and show them to you. But Helen did a great job during a transition in her life and a transition for all of us. So thank you, Helen. So DIRT, soil. Soil is arguably one of the most diverse entities on Earth. A teaspoon of soil contains more organisms than there are people on the planet. Soil is a mixture of fungi, bacteria, archaeobacter, worms, collembola, centipedes, millipedes, all kinds of organisms that chew up and digest and exude chemicals and compete with each other in a marvelously complex system that we're only beginning to understand. When I say we, I mean science is beginning to understand. This is a graphic from the New York Times article, 2002, I think. I can't see my own slide. Yeah, 2002. An article where E.O. Wilson, Harvard biologist, expert in biodiversity, pointed to soil as being the most complex system on Earth. So take a good look, and then We'll talk about it. We'll talk about the biogeochemical controls on forest soil organic matter accumulation. Lessons from the dirt collaboration. So those of us who study ecosystems often refer to ourselves as biogeochemists because we have the challenge of linking biological processes to geological processes to chemical processes. And again, this takes tremendous collaboration, and those collaborations have to be successful if we're going to learn anything. Why are soils important? Why do they rule? Well, they're habitats for organisms, as E.O. Wilson explained in 2002. But they also sequester carbon. So soils contain more than twice as much carbon as the atmosphere contains. And if we can keep soil, if we can keep carbon in soil, we can partially offset the increase in CO2 that's resulting from our fossil fuel emissions. And if we can store more carbon in soil than we emit from soil, we can actually offset some of that carbon. So there's a lot of talk now that we're entering the climate crisis about carbon sequestration, and a lot of that talk is about engineering for carbon sequestration, big vacuums that suck CO2 out of the air, but plants and soils together do that as well. And in fact, northern forests in the northern hemisphere remove about 1/4 of all human emissions of CO2 from the atmosphere. The oceans remove about another fourth. So if the forests and the lands and the oceans together weren't removing carbon, CO2 would be increasing twice as fast as it is now. So they're providing a service, which is climate regulation. They also, and unfortunately, I'd have to give a class in this, but along with all that, carbon cycles nutrients, nitrogen, phosphorus, calcium, potassium. And so that carbon regulates those nutrients cycling along with the organisms. The water we drink from our wells is purified by the organisms that break down compounds before the water can trickle down into the water table or leave a catchment into a river and a lake and an estuary from which we get water. Flood regulation, the organic matter in soils, and the thing I love most about soils is organic matter. Organic matter holds water, it holds nutrients. It's critical in flood regulation. And when people restore wetlands that are rich in organic matter, they're regulating floods. They're diminishing the intensity of floods. They provide, of course, food, fiber, fuel, and they're a source of pharmaceuticals. Penicillin was discovered in soil, many others. All the organisms that are fighting each other and collaborating with each other do it chemically, and they release chemicals that sometimes have pharmaceutical value. So there's a whole science looking for pharmaceutical compounds in soils. Also, soils hold much of our cultural history. The land acknowledgement that Director Clausen made relies a lot on what people did and how they used the land earlier. And our soils at the biological station and around the biological stations have cultural artifacts, they have cultural value. And I'm happy that the biological station and the University of Michigan have been able to protect over 10,000 acres around Douglas Lake so that those archaeological treasures are not lost by development. And also we build stuff on soil, but that's the least of my interests in this talk anyway. I like to build stuff, but I like the living things in soil. So soils are the topic of many workshops. Amy and Luke have been to them. Luke has organized soil workshops. And these are the drivers of the carbon soils, SOC, soil organic carbon, microorganisms and fauna, that diverse community. They're processing all that material. Earthworms will eat leaves, leaf litter whole, grind it up in their guts, mix it with sand and clay, allow the microorganisms to digest it, metabolize the carbon. Texture, the amount of sand or clay or silt in a soil can control storage. The way we manage land, how we turn the soil over, how we crop it, what kinds of trees we plant on it affects carbon storage. The vegetation, in terms of what it donates to soil, controls soil. Leaves fall, we call it litter. Roots die, we call it root turnover. Roots exude carbon to feed bacteria and fungi so that they can respire, produce enzymes to release nutrients for root uptake. Topography, flat topography, mountainous topography, water runoff, that controls soil organic carbon. Parent material, what kind of minerals the soil is derived from, and then soil physical chemistry, all the chemical soup that these organisms are producing affects soil carbon. If we look at how carbon is partitioned in soil, the atmosphere, and in vegetation, both biota and detritus, which was basically dead leaves and dead roots, the smallest of those pools is the vegetation pool. About 500 billion metric tons of carbon are in vegetation worldwide. Almost 3 times that amount are in soils, and this is only soils to a meter depth, and it does not include tundra soils and permafrost soil, which have even more carbon. And the atmosphere is, about a little over 2.5 times, or excuse me, a little less than half of soil carbon, a little more than half of soil carbon. So let me explain this. So all of that carbon that's in soil, topsoil and subsoil, and this is again the top meter, is there's about, if we add up these bars, there's about 400 billion metric tons in temperate forest soils. If we go, and almost, very little in biomass. Actually, if we want to store carbon in soil, in forests, in the temperate zone, we want to ultimately store it in soil where it lasts a lot longer than it does in vegetation. But we have to grow the trees to capture the carbon, to throw the carbon into soil. Tropical soils, or tropical forests, on the other hand, are rich in biomass carbon and fairly rich in soil carbon as well. And then boreal forests, the northern forests of Canada, the Soviet Union, some in the US, and Alaska contain a lot of soil carbon and a little less vegetation carbon. Now, but biogeochemists, ecosystem ecologists love models. This is the simplest model I could come up with. We have these 3 slices of the pie: vegetation with about 500 billion metric tons, atmosphere with about 875 billion metric tons, and soil, surface soils with about 1,400 metric tons of carbon. Just go back here. Atmosphere and the vegetation exchange carbon. We all know about photosynthesis. Photosynthesis pulls carbon out of the atmosphere, stores it in vegetation, but plants respire just like we do, so they send back CO2 to the atmosphere. On balance, they're pulling more out of the atmosphere than they're sending back in respiration. That's why plants grow. Vegetation also donates carbon, donates litter, roots, bark, all kinds of plant tissues to soil where it decomposes. And soil, when it decomposes, the organisms that are in the soil release CO2 when they respire. So soil is a two-way street. It's taking in carbon from vegetation, but it's donating it back to the atmosphere. If the inputs from vegetation are equal to the outputs in soil respiration, then soil's carbon sink is 0. But if the inputs from vegetation to soil are greater than soil respiration, then soil is a carbon sink. It's a positive carbon sink. And if the transfer of carbon from the soil to the atmosphere is greater than the transfer of carbon from vegetation to the soil, then it's a source. And I'll give you some examples of where we had sources. But before I do anything else, I want to tell you about, this is a story. I want to tell you about how this dirt network originated. And it starts at the University of Wisconsin Arboretum in 1956. In 1956, Francis Hole was a young professor at the University of Wisconsin. John Curtis, a plant ecologist, had established the University of Wisconsin Arboretum to be a new kind of arboretum. It'd be an arboretum where ecosystem types across Wisconsin were replicated. So an old farm was bought on the outskirts of Madison. Cornfields were turned into prairies. Woodlots were turned into oak forests, maple forests, pine forests. Francis Hole started an experiment then, but I want to talk a little bit more about Francis. Francis not only studied soils, he was an amazing person. He lived on the east side of Madison. We took the same bus every day when I was a PhD student. And Francis was a simple person. He was a Quaker, he was a pacifist, he was kind, he was knowledgeable about soils. He worked with the Menominee tribe in northern Michigan to map the soils in their fairly large reservation. He's the person who first introduced me to Native Americans and their respect for the soil, as well as the other resources that they managed and still manage. So Francis was an amazing person. He loved kids. He loved talking about soils to kids. He loved making presentations, and he wore a badge that said Francis Hole TNS. And when the students and the kids would ask him, what does TNS mean? He would say, temporarily not soil. I'm Francis Hole, temporarily not soil. I just thought that was amazing, that someone could be that casual about mortality. The lesson I'm learning more every day. Francis described soils with music and songs. He described soils with a violin. He'd say, this is the surface soil, and he would play, something like a surface soil. Let's see, I just have a little thing. In fact, Francis composed songs, and I hope I can, I don't know if yeah, I can. Okay, Francis translated an Italian song, Viculi, Vicula, and I'll play it for you as soon as I can move this little bar. And then we'll look at his translation. I don't know if it's accurate, but we'll see. Now, that was written by an Italian composer in the 19th century to commemorate a funicula that was built up Mount Vesuvius. Of course, volcanoes are where soils originate. So Francis decided he needed to write words to this song. So follow along with Luciano Pavarotti for the first few verses and read Francis's translation. I will not sing it for you. I don't have my guitar. This is not true. This is not true. She gives us bread. Okay, enough. You get the point. He was an ambassador for soil, and he was an artist and a scientist and a humanitarian. A man I truly, truly respected and loved. We would ride the bus together to the university in the winter. Francis was such a simple person. He would carry his books in a shopping bag. He would wear the kind of yellow gloves you can buy at the hardware store when you're gardening as his winter gloves. And Francis and I would sit in the bus, and he would tell me all about soils. And so and he wore an old, coat with holes in it and everything. And I just can't imagine what the people around us in the bus were thinking when we were talking about soil. But he told me in that bus conversation about an experiment he started in 1956. Oh, let's get back to art for a minute. So this artists have been representing soils forever. This is by Van der Weyden in 1432. If you look at the bottom, you can see people emerging from soil. Dusts in me, dusts in you. If there were soil scientists, then they were consulted on how the soil profiles were represented. Grant Wood, who you may know from Iowa, who wrote, who painted a picture with two farmers, a man and a woman in front of the barn, American Gothic, also painted soils. Here's a soil profile in 1932. Now, if you remember 1932, the 30s were Dust Bowl days. These soil profiles were obliterated. Farmers were planting fence post to fence post, moldboard plowing. Soils were mismanaged, and they basically resulted in the Dust Bowl. This is the Dust Bowl material. This is the carbon when Europeans colonized North America. They were happy to plow up and plant corn and plant food. And that a lot of that soil was spired away. The inputs were less than the outputs to the atmosphere, and we lost carbon from the breadbasket of the US. So as in the Depression, the University of Wisconsin Arboretum was created, 12,000 acres, about 1/10 the size of the biological station, but still a lot of research was done there. These are the different communities, and I'll just very quickly talk about two of them, Noe Woods and Windgrove Woods, both oak forests, both places where Francis Hole, there he is at the Arboretum in a prairie, developed the first dirt experiment, studying the natural processes of the incorporation of organic matter into soil at the University of Wisconsin Arboretum, Wisconsin Academy of Arts and Sciences. Francis gave me a copy of this. That's my handwriting right up there. And this is his experimental design. He said, John Curtis asked him to start a soil experiment at the Arboretum. And he said, well, you know, I think soil really requires litter to develop. And so I'll look at some soils. This is my control. This is topsoil with litter doubled. And how do you double the litter? Well, you rake the litter off of a plot and you double it up on the other plot. And then just to simulate what happened in the Dust Bowl, he removed all the topsoil from the soil, replaced it with subsoil, and then just let nature take its course. A brilliant experiment, I thought. When he retired, the director of the Arboretum asked him, how long should we keep this experiment going up, Francis? And he truly said, oh, at least 1,000 years. And I thought, well, that's an ambitious science project. So we have a simple experiment at the University of Wisconsin Arboretum. And I went back when I was a postdoc in Woods Hole and sampled in 1984. The experiment continues through today. And rather than show you data, I'll show you some art. These are mixtures of soil from the different treatments that I collected in 1984. The control with normal leaf litter inputs looks like this, very dark and rich in soil organic carbon in the upper 10 centimeters, a little less lower down, a little less lower down than that. I don't have pictures of below 20 centimeters. Plots from which litter were excluded are lighter in color, less organic matter, lighter in color even in the lower horizon. Double litter, can't really tell, but it's a lot darker. The data will show more carbon here. And the A less, this is the recovering soil with natural inputs after 28 years, 56 to 84, is about 70–75% recovered. A little lesson in restoration ecology. These are the people when I was in 1997, went back to sample, and finally we got Francis Hole to come with us. Rich Boone, who's just retired from the University of Alaska Fairbanks, Pat Mix, who helped with the dirt project at both Harvard Forest when I started it there, and at the University of Michigan Biological Station is here, and Jana Canary, a student at the University of New Hampshire, also helped us. I don't know who this is, my memory fails me, but he guided us to the plots. And Francis Hole, of course, was here. And Francis was, you know, he was one of the soil scientists who hated soil to be referred to as dirt, but he got the acronym and he had a sense of humor, so he didn't hold it against us for developing his model as a bigger dirt experiment. Here is Francis in the field approving our dirt experiment with the 3 people I just mentioned at the University of Wisconsin. And here is some simple data. This is Francis's experiment started in 1956. Here's the last sampling, which was in 2004, I think, 97, and here's 84. So you can see the double litter experiment has about 25% more carbon per gram of soil and surface soil, and about a little less than half when litter was excluded. That was at one of those plots, Noy Woods. The other plot, a similar pattern, a little smaller magnitude of increase in carbon. So I love decomposition, and this is my compost pile. And I loved it when I was a kid, and I never quite understood why when you put all this kitchen waste in a pile, it got smaller. Well, again, it's the organisms that are processing it, whole cadres of organisms, whole mixtures. So this stuff I love. I just love throwing stuff in there and watching it disappear and turn dark and brown. Let's build a little bit more of a complex soil model. The pie chart that I showed, which is 3 wedges. Here's the vegetation wedge. Remember, it takes carbon from atmosphere. It removes carbon from the atmosphere. It returns it via respiration, but also via litter fall, by changes in leaf litter, fine roots, bark, wood, buds, flowers, fruits, bracts, all kinds of stuff, including exudates from roots that feed microbial cycles, exudates of carbon-rich sugars, and miscellaneous things. So we can think about soil as having fresh inputs, like in that compost pile you saw. The carbon to nitrogen ratio is high. There's a lot of energy in that material. That energy subsidizes, drives those microorganisms and all the soil organisms. As the soil gets processed, carbon gets respired to the atmosphere, but some of that carbon gets stored in compounds that have lower percentages of nitrogen or higher carbon to nitrogen ratios. That gets respired by different suites of organisms. Some of that goes to the atmosphere. Some of that goes into humus, which has a carbon to nitrogen ratio about the same as bacteria. It's almost pure amino acid, nitrogen-rich compounds, has very little energy in it. So these organisms that decompose this energy-rich material will also mine this stuff, but they'll do it at a carbon cost. All of these conceptual pools release carbon to the atmosphere. And when the sum of, as the model before, when the sum of the vegetation to soil transfers is greater than the sum, or is equal to the sum, is greater than the sum of soil to atmosphere transfers, soil removes carbon from the atmosphere. It stores it, it's sequestered. When the sum of soil to atmosphere transfers is greater than the sum of vegetation to soil transfers, soil loses carbon. So our challenge is to develop systems and to understand how we can alter the balance so that more carbon is put into soil than is released. So let's go back to the dirt sites. University of Wisconsin, established 1956. When I moved to Woods Hole, I took Francis's idea and established a dirt site at the Harvard Forest in 1990. Rich Bowden was a postdoc in Woods Hole, and when he left in 1991, he established a dirt site at a maple forest in Western Pennsylvania called the Busan Forest. Our friend Kate Loida at Oregon State established a dirt site at the H.J. Andrews Experimental Forest, 1997, and then we had European friends, and then in 2004, the Biological Station established a dirt experiment. Others around the world have been started. I can't keep track of them all. Here are 2 that are in dry lands and forests, mountainous forests. Now, we took Francis's experiment and we made it complicated, and here it is. I'm not going to talk about data from all this. I only have a little data to show, but we have a control plot. We remove litter from no litter plots, as Francis did. We take that litter and we make double litter plots. We also fertilize some plots and we add double litter to the fertilized plots, and we just fertilize plots without adding double litter. We created root barriers. We dug trenches to exclude roots. And then we didn't let anything fall in the soil. We took off the leaf litter and we had root barriers. So we have control, no litter, no roots, no inputs, and double litter. That's what I'm going to focus on. And let's go back to Wingrove Woods and Noy Woods and the Arboretum. This is the double litter compared to the controls. This is the no litter compared to the controls, and you can see pretty clear increases. Those are significant. I don't have any statistical symbols on this, but you know, as I said, 25% or so of more soil carbon in the double litter, and here about half as much when litter's excluded. So inputs matter. The Harvard forest, which is on a sandy soil, like the biological station, had a control, double litter, no inputs. You can see there's no increase in double litter in the top several centimeters of soil. There's no real decrease in the no inputs, a little bit of a decrease in the no litter, no roots, no decrease. And the OA, we created an OA there. We removed the surface horizon, replaced it with subsoil. And after 20 years, it hadn't recovered even 1/3 of its original carbon. The Busan experimental forest, which is very much like Harvard forest, which has fine textured soils with a lot of clay, no inputs of litter, lost carbon, double litter. Surprisingly, we thought it would increase in carbon. It didn't after 20 years. No litter, a little less decrease than no inputs, and no roots, not much of a decrease. The H.J. Andrews forest, a similar pattern to Busan, and that's a fine textured volcanic soil. So texture matters. When we sample the plots, we sample the profiles. So we take what we call a brownie. This is the organic layer off the top, and sequential soils at 10 centimeter increments. I'm going to go fast here. We have collaborators who study these things: enzymes, fungal, bacterial communities, chemistry. We measure the pools. So this is a soil profile. What we'll call the OE is that organic horizon up here. The A is here. We combine the OE and the A, and we sample that. We analyze it for carbon, nitrogen. We measure its bioreactivity, and we do the same with the different depths through the profile. 0 to 10, 10 to 20, 20 to 30. This is Richard Spray, one of our biological station staff members, helping us dig trenches around plots with Jim Lemoyne. These are the dirt plots being established in 2004. Root barriers, soil all around, plastic to prevent soil from going onto the plots. These are 5 by 5 meters. This is what a plot looks like when it's finished. All kinds of instrumentation to measure things in the field. So this is a no litter plot. Litter is raked off and removed, distributed to the double litter plots. This is how we sample. We take a brownie. We take depth increments with a soil core. And this is the OEA horizon. Again, this stuff. Beautiful stuff. Notice these are the sequential years. 2004, 2005, 2009, 14, 19. This is a surprising and interesting and troubling thing. The carbon content of that horizon at the biological station over 15 years has decreased from almost 1,000 grams carbon per meter squared in soil to just over 550. We've lost almost half of that OEA horizon without doing any experiments. The no litter started out lower, and continued to lower in its carbon content. The no roots, similar pattern, and the no inputs, similar pattern. Overall, when you compare any individual treatment across years, you do see some changes as well, but the overall treatment, the overall decline in soil carbon is troubling, and it may be the result of earthworm invasions. It may be a warming climate, which is increasing metabolism of the organisms. We just don't know. Double litter did not suffer that decline. This is year 1 and year 2, so I think that's an artifact of sampling. Fertilized, I really won't talk about that. We don't have time, but we did fertilize plots as well. When we went down the profile, carbon increased. So a lot of that carbon that's coming out of the top layer of the soil is going into mineral soil, increased across all the treatments. So let's just look at a couple of treatments. Let's compare the control with the no input. Nothing at all going in, no roots, no leaves, root barrier, that stuff's being taken away. The control soils are the comparison. And in 2004, they were more or less the same. These are standard error bars, these are means. By 2009, let me explain this. This is the OEA. This is the 0 to 10 mineral soil, and this is the 10 to 20 mineral soil. This is the carbon stock in grams carbon per meter squared of forest soil. We can see the no inputs by 2009 were considerably less than the control, and most of that loss was in the OEA, the top brownie horizon. Some of it was in the 0 to 10, and a little bit of it was in 2009. So when you remove inputs, soils get impoverished. And 2009, the story is the same, except the OEA horizon is almost entirely gone after 15 years. So those are the extremes. This is the data I just showed you. I'll show you one more extreme, control and double litter. So 2004, O horizon with double litter, 0 to 10 centimeter and 10 to 20. This was larger then. Both of them had increased by 2009. We were surprised that this OEA wasn't greater, but metabolism clearly kept it down and transferred some of that carbon to lower soils, but it didn't have as much carbon as we expected. 2019 was a little bit of a different story. Still, there was no more in the OEA horizon there is in the O horizon in the control, but a lot of carbon had been transferred to this lower layer. So think of that model where the high carbon to nitrogen leaf litter is being transferred, being eaten up, digested, and the byproducts are going lower in the soil. They're being transported down by rain and moisture and organisms to accumulate in the next layer of soil. I'm not going to show fertilizer, we don't have time, but I'll also show you data from the lab. We do so many lab analyses, and really this is the surface of the experiment, literally. There's a whole lot more data. In fact, this is going to be 2 or 3 years of my emeritus status that I'm going to be analyzing data from this sequence and producing some really cool papers with my really cool collaborators. This is a apparatus that we invented back in Woods Hole to measure soil respiration. You put soil in a little blood serum cup counter. You measure the CO2 that comes out the top. You pour solution in here and you measure the nitrogen and the dissolved carbon that comes out the bottom. And this is an array of them from different plots and different treatments in our lab in Michigan. And this is an example of how much CO2 carbon per hectare equivalent was respired from those laboratory mesocosms. So I have to move so you can see that even though the pools are, the amounts in the field are not always so different, sometimes are, sometimes in different soil horizons, they're accumulating, whereas they're not in the upper. But if you just look at the upper soil horizon, the organic soil, the respiration, the CO2 produced in the double litter is way higher than the CO2 produced in the control. The no litter, less respiration, no roots, less respiration, no inputs at all, hardly any respiration at all. And then when we fertilize and double litter fertilize, we get huge respiration. So summary, the soil surface horizons at the UMBS dirt site are losing carbon through time. Surprise, surprise, why is that? There's a whole research program that could be built around that. Surface horizon carbon accumulation varies with amounts and sources. That is, the amount that goes in or doesn't go in, and the source, whether it's above ground or below ground, more or less as we expected. Much of this carbon is being transferred to deeper soil layers. And, we need to go deeper than 30 centimeters because these soils are sandy and porous. And I think a lot of the carbon is accumulating down there. Surface soil horizons and deeper layers respond differently to amounts and sources of plant inputs, as those last graphs showed. And soil biota, as illustrated in the incubations in the lab, are highly sensitive to amounts and sources of plant detrital inputs, roots and leaves. And we really have to understand how they're transforming compounds, what those compounds are, how resistant they are to decay, how much nitrogen they hold. And we have built a whole research program to look at that. And the collaborators that are some of the people looking at these more micro scale processes are busy at work trying to tease all this out. So we have much to learn. And I see that some comments have been made in the chat box, and I'm ready to thank you all and entertain any questions you have. I'm sorry if I ran a little longer than I should have, but if I used all of our data, we would be here for weeks. So thank you.
Thank you, Knute. I'm piping in now. This is Jenny. I'm the communications coordinator for UMBS. I don't know if you can see my video, but I'm coming in live from the UMBS store under the harsh fluorescent lights in here. May look familiar to people who've been to the station. So now if anyone has questions for Dr. Nadelhoffer, I'm happy to read those aloud to him. So please ask them in the Q&A. And I see we've got a question here for you, Knute, from Jacob. So were the plots covered with plastic the entire time? Is sunlight not that important to the organisms and the processes involved? And what about rainfall and moisture? Wouldn't the centers of the plots receive less moisture given that the plots are covered? Or does moisture not affect the carbon processes significantly?
Wow, that's a lot of questions, but they're good ones. Yeah, what I failed to mention is that we actually normalize these treatments. So what we do, and with work from Jim and John and a whole lot of students, we remove ground layer vegetation from all of these treated plots so that all they get are basically leaves and roots from trees. The reason for that is when you double up litter or remove litter, you actually create different conditions for ground layer vegetation, and you're all of a sudden changing ground layer vegetation in ways that you don't understand, right? So we basically remove every year, we pull plants out of those plots. The plots that don't have any leaves develop moss, and we actually pull that moss off. So we've had lots of students and myself and particularly Jim and John who do that work all the time. So we've normalized that. Okay, so the only thing that's really differing are tree inputs to the plots. We do measure moisture. We actually have what are called Lysimeters, soil water samplers. So we measure the amount of water the chemistry of the water that trickles through the profile. Every time we take a sample, we measure the amount of water in it. And you're right, when a soil has more organic matter, it has more water. When it has less organic matter, it has less water, because that organic matter, those fine little pieces, create surfaces that water can adhere to. And so we're not just changing the leaf layer inputs, we're changing the conditions that the litter and root inputs create. So we can't really single out, roots alone or leaves alone, but we can begin to understand how the leaf litter and the root litter affect everything that has to do with soil carbon storage, including organismal respiration, including moisture content and temperature. And so the temperatures of the, when you put twice as much litter on a plot as control, you change the temperature. And we have temperature sensors that do that. And we take temperature whenever we go in the field, but we instrumented the plots. So there were all kinds of snake wires going out, measuring soil moisture and soil temperature. So we do try to measure those variables, at least to develop statistical models that can look at covariance between temperature and moisture and the things we're measuring.
Wow. So Knute, there's that half man, half genius that Luke mentioned, because you just anticipated the next question. So maybe you have something to add to this, but Steve Bertman, who I know is watching with the RU students here in camp, asks, do you have temperature measurements at the different layers of soil?
I wish we did. We have temperature measurements just a little bit below the OA horizon, I think about 15 centimeters from the surface of whatever's there. We have thermistors, but we just didn't have the resources to do full profiles. Be a good thing to do. We could do it.
Okay, we've got a question from Dr. Luke Nave, your illustrious introducer. So Luke says, soils are clearly very important to our survival, yet we've made little traction as a scientific community in getting a quorum of people to care. Can you tell us an example of a way that you've gotten through to someone about the importance of soils or of this experiment?
Well, my model for getting through to people is Francis Hole, an ambassador for soil. And I could tell a story about that. That's a challenge we all have, Luke. That's a really good question. You've been really good at this because you've headed up soil carbon efforts. You basically, well, you could explain better than I could what your role has been, but you've been coordinating for soil carbon measurements all around the country until very recently, and that's really valuable. So, you know, I think articles like the New York Times article about soil carbon, I think the emerging climate crisis it sounds funny, but it's an opportunity to talk about how important soils are, not only for food, not only for you know, the other things we clearly see soils being valuable for, but climate regulation. You know, the more attention people are paying now to floods, intense storms, stronger and more frequent hurricanes, fires resulting from drought and heat waves in the West, but also other parts of the world, Greece. Those are all opportunities to bring people into soil. I mean, when the general public, many of the people in the general public think about biological carbon sequestration, they think about trees. But really, a soil pool is a longer turning over pool. It's a more resilient pool to change than a tree. A tree can be cut down, a tree can burn. Soils, they respond to that kind of disturbance, but they respond much more slowly, and they typically have more carbon than the trees above them, except in the temperate forest or tropical forest. Yeah, so I could say, so an example of Francis Hole. Are there more questions?
Yeah, we've got a couple more questions for
Okay.
you.
I could tell a Francis Hole story, but I'll defer to other. Francis was a great ambassador.
Yeah, tempting.
Oh, here, okay. You know how everybody has, all states have like a state bird, a state flower? Francis Hole pioneered the naming of a state soil in Wisconsin. So every soil, you know, profile, as soil scientists know, has a different name. And Francis, you know, depending on how thick the soil layers are, what their colors are, how much stuff they have in them, Francis was expert at that. It's called soil taxonomy. And he pushed in the Wisconsin legislature to have the Antigo silt loam as the Wisconsin state soil. And since then, well, anyway, that was a great idea. And he got a lot of press for that. And I think Michigan has a state soil. Luke would probably know. I should. And
Kalkaska, isn't it?
Now do at least 30 states have, you know, state soils. So there's an opportunity. Anyway.
Thanks, Knute.
Yep.
So we do have a few more questions for you. One from Dr. Christopher Goff, who's a dear friend and collaborator here at the Biological Station. And he says, great to see you, Knute. Such an important example of the value of long-term ecological data. My question, how did available nitrogen in the soils change in response to treatments?
Well, Yeah, at the risk of prolonging the talk, I took those slides out, but available nitrogen basically changed with carbon. I mean, especially the nitrogen that's rolling out of the carbon. So when you collect soil from a plot that's been enhanced with litter, the nitrogen release from soil in the lab increases pretty dramatically, actually, because that fresh stuff, that energy source is increasing, plus there's more nitrogen coming in from the litter, plus the roots are actually growing into that nitrogen, turning over faster, so it gooses the whole system. Then, and this, the inverse happens when you remove input. So nitrogen follows carbon, but in funny ways. When the respiration of the soil is high, the nitrogen release is low, but as the soil in the laboratory starts to get processed, the nitrogen flows out of it. So really, when that fresh carbon comes in, when that high seed end litter stuff comes in the fall and winter, it's retaining nitrogen, but as it gets worn down, a lot of the bacteria die, get eaten, and they release nitrogen. So that there's kind of a neat interaction there that I wish I had time to show some figures, but you can see the carbon increasing and then going down. And as the carbon emission, the CO2 emission goes down, the nitrogen goes up.
Thanks,
And the
Knute.
magnitude of that difference is roughly in line with the amount of respiration.
We'll ask you two more questions before we wrap up tonight. One from Elliot, who was a UMBS student this past summer. Thanks for tuning in, Elliot. And Elliot says, 1,000 years may have been an exaggeration, but do you expect to keep getting novel results from this experiment over many decades?
I do. The problem will be separating it from the climate effect. But at least we can see how these different treatments are responding to a change in climate, which is happening, So Kate and myself and Rich Bowden, we're all I'm the first one to sort of retire, and Kate and Rich are retiring. I think AMA has agreed to keep the UMBS dirt experiment going, and all we can do is say, okay, keep it going, keep measuring. If it starts getting boring, then stop. But I should mention too that most of us have done this without any major grant funding. It's just something we all love to do. We love to play in the dirt, and we get little sources of resources here and there, and sometimes someone gets an NSF grant, but we have never had a huge grant that could power drive all these experiments. So that the next person, a brilliant and artful person, needs to write that proposal. So I hope there are students here who are gearing up to do that if they're interested in this.
And we'll ask one final question of you, Knute, from Steve and the REU students. Do you think that the DIRT project could help inform or encourage regenerative agriculture practices on a large scale?
Yes, absolutely. Now that you ask those two questions, there's an old experiment that is actually centuries old by now in the UK, in England, called the Rothamsted experiment. And that is exactly, aimed at addressing that kind of question, where cropping systems have been varied in terms of how much and what kind of plant waste, really. I mean, waste in terms of non-edible stuff, straw and, chaff and things are added to agricultural soils. And, we already know something about that from no-till and minimum till and agriculture, but there's still, and our colleagues at Michigan State have a whole long-term ecological research program that's designed around sustainable agricultural practices that aim to put carbon in soil. And so I think, the more these practices can be shown to be keeping soil carbon in the soil, adding soil carbon to soil, the more we can develop incentives for farmers to continue those practices through, government subsidies and better crop yields and less fertilizer. So that's a really good question. I hope I answered that. I don't know. I sort of got off topic maybe, but.
That was great, Knute. And with that, I think we'll wrap up tonight's talk. And so I, of course, want to thank you very, very much for a wonderful talk, as we all knew it would be. We're so lucky to have you back for this Endowed lecture. And thank you for your time. Thank you, everyone who tuned in on behalf of the Biological Station. And you'll just have to reach out to Knute with all your bonus questions here. So thank you, everybody. And thank you, especially Dr. Knute Nadelhoffer, for a wonderful talk.
Well, thank you all for bearing with me for an hour and 5 minutes, but I really enjoyed talking and I love the questions. And yeah, thank you for the invitation, everyone.
All right. Thanks, everybody. Take care.
Bye-bye.